Hello, everyone, and welcome to today's Session 3 webinar, Simple Steps to Publication Quality Western Blots. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. Now, we encourage you to participate today by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. Now, this webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd now like to welcome our speaker, Kelli Feather-Henigan, a Senior Product Manager at Protein and Cell Analysis at Thermo Fisher Scientific. Kelli, welcome. You may now begin your presentation.
Hello, everyone. Thank you for joining Part 3 of our webinar series, Simple Steps to Publication Quality Western Blots. My name is Kelli Feather-Henigan, and I'm a product manager at Thermo Fisher Scientific supporting our Western blotting products. Before becoming a product manager, I was an R&D scientist for over 15 years, and during that time, I performed many Western blots and developed many of our current Western blotting products. As you may know, in recent years, there's been a growing concern regarding the reproducibility of and quality of results in Western blotting data submitted for publication in peer-reviewed journals. In 2014, the journals of Science and Nature, along with the National Institute of Health, organized a meeting with other publishers, journal editors, and scientists to discuss reproducibility and transparency of published scientific data. The concerns raised over reproducibility and quality of Western blotting data submitted to peer-reviewed journals led the meeting collaborators to revise their instructions and recommended best practices for submitting non-quantitative and quantitative Western blot data for publication. This webinar series has been broken into 3 parts. Today, I'm presenting Part 3 of the series, which will focus on improving reproducibility in Western blots and special considerations for obtaining quantitative Western blot results. Demonstrating reproducibility of Western blot results has become a focus of some publication guidelines. Anyone who does Western blotting will be very familiar with the workflow depicted on this slide. Western blotting is a very large, labor-intensive workflow with lots of hands-on steps and areas where variability can be introduced. With any process that consists of several time-intensive, hands-on steps like a Western blotting workflow, small inconsistencies at any step can lead to larger issues of blot-to-blot consistency, which could impact the reproducibility of results. Today, I'll be focusing on measures that can decrease the amount of hands-on activities in the Western workflow and can lead to improved consistency and reproducibility across blots. Invitrogen's modern Western workflow helps minimize hands-on steps and reduces time to results compared to the traditional Western workflow. The electrophoresis in steps 1 and 2 can be simplified using our Precast gels and gel tanks. Step 3, the transfer step, can be simplified and time to results reduced by using our iBlot 2 instrument and iBlot 2 pre-assembled transfer stacks. The time required for transfer with the iBlot 2 system is reduced to 7 minutes or less, and the iBind device shown in step 4 of this slide can reduce hands-on time by automating the Western blotting immunoassay steps. Image capture and results analysis can be simplified using our iBright digital imager. Precast gels can be used to greatly improve reproducibility of Western results. Precast gels have many advantages over manually poured hand-cast gels. Precast gels have better consistency because human error has been minimized and the gels are backed by quality control standards. They're conveniently ready to use, so they have save significant time by eliminating the hand casting steps. And our precast gels come in a wide selection of chemistries and sizes for our mini gel tank and our Sherlock tandem midi gel tanks, both shown there on the right. We have standard tris glycine and tris gels in a broad range of gel percentages and gradients. Tris acetate gels, which are especially great for separating large molecular weight proteins. Tricine gels, which are ideal for separating small molecular weight proteins, and a variety of other gel chemistries to meet other needs, such as native page gels and isoelectrofocusing gels. Our iBlot 2 rapid dry transfer device shown here can be used in combination with our iBlot 2 pre-assembled transfer stacks to minimize handling inconsistencies in transfer steps, and it reduces time to results by transferring protein from gel to membrane in 7 minutes or less. The iBlot 2 system has a built-in power supply and intuitive software that can be easily programmed to successfully transfer protein across a broad range of molecular weights from most types of SDS page gels. The iBlot 2 transfer stacks come ready to use with either nitrocellulose or PVDF membrane, depending on your preference, and they come in both midi and mini sizes. The iBlot 2 system can transfer 2 mini gels or 1 midi gel at a time, and because the transfer stacks are pre-assembled and contain everything needed to transfer proteins from gels to membranes, results are more consistent and hands-on time is reduced. The iBlot 2 system provides consistent transfer across a wide range of gels and gel chemistries. These data show the rapid transfer of protein using our iBlot 2 system with iBlot 2 pre-assembled transfer stacks, and here you can see efficient transfer of high molecular weight protein from 3 different gel types and on both nitrocellulose and PVDF membranes. Another option for improving consistency in Western blot transfer is the Invitrogen Power Blotter semi-dry transfer system with pre-made transfer stacks. The Power Blotter system enables rapid transfer of proteins from a broad molecular weight range in 10 minutes or less when used with our Power Blotter one-step transfer buffer. But it's also versatile and can be used with conventional semi-dry transfers with Tobin transfer buffer. Power Blotter system provides the flexibility to choose between nitrocellulose or PVDF membranes in our ready-to-use transfer stacks that contain everything needed to perform rapid transfers, or we have ready-to-build transfer stacks that can be combined with our Power Blotter one-step transfer buffer for rapid blotting in 7 to 10 minutes. Those ready-to-use transfer stacks or ready-to-build transfer stacks can also be used with standard Tobin transfer buffers. The Power Blotter system can be a higher throughput option for transfers because it can be used to transfer up to 4 mini or 2 midi gels using the Power Blotter system with the XL cassette. Using pre-made, ready-to-use transfer stacks can increase efficiency and reproducibility of Western blot results. The blots on the right show an example from a transfer proteins using one of our ready-to-use stacks versus a traditional build-it-yourself transfer stack. On the bottom membrane, you can see an example of an inconsistent transfer. Handling inconsistencies can occur when building traditional do-it-yourself transfer stacks. Things like uneven membrane hydration, insufficient removal of methanol or ethanol from PVDF membrane, and stacks drying out while other stacks are being built can cause splotchiness and uneven transfer. Incorrect setup of stacks with too few filter paper layers or incorrect membranes or gel orientation can also cause poor transfers and inconsistencies. These data show the reliable blot-to-blot transfer performance that can be achieved by decreasing handling inconsistencies. In this example, 12 individual Bolt 4 to 12% Bistress Plus gels were transferred using the Power Blotter with the Power Blotter Select stacks, and each blot was probed for the same target protein. The transfer results are very consistent across all 12 transfers, demonstrating how the elimination of handling inconsistencies will allow you to provide images to your publishers that are more representative of all of your experiments. You can increase consistency and decrease tedious hands-on steps in your Western blots by using an automated Western blot processing device, such as the Invitrogen iBind Flex Western device. The iBind Flex allows for automated immunoprocessing. You simply load your reagents into the reagent reservoir in the system, and the iBind device utilizes sequential lateral flow to perform the immunoassay without electricity, shakers, trays, or timers. The iBind device provides consistent, robust results, often with less primary antibody than is required with manual processing methods. The example on the right shows a fluorescent and chemiluminescent signal from blots processed either manually or with the iBind Western device. The results are equivalent or better using the iBind system with 80% less primary antibody than that used in the manually processed blots. Here's another example of results with the iBind Flex system. In this comparison, Western blots processed manually or with the iBind Flex Western device are shown. Samples containing GST-tagged recombinant proteins were separated on Invitrogen New Page 4 to 12% gels in MOPS SDS running buffer, and then these were transferred using the iBlot 2 gel transfer device and stacks. The blots were probed with identical concentrations of the same pair of primary and secondary antibodies. The primary antibody was rabbit anti-GST diluted 1 to 500 or 8 microliters in 4 mL of iBind Flex solution for the iBind system, or 40 microliters in 20 milliliters for manual tray incubation. The secondary antibody was goat anti-rabbit HRP diluted 1 to 600 or 6.7 microliters in 4 mL of diluent for the iBind system, and 33.3 microliters in 20 milliliters for the manual tray incubation. For final detection, blots were incubated for 5 minutes in Supersignal West Dura extended duration substrate for visualization on an imaging system. These results show the consistency and reproducibility of Western blot results using the iBind Flex system with less antibody compared to manual blot processing. The iBind device comes with interchangeable wells that allow you to process multiple blot formats. There are iBind Flex well inserts that will allow you to process one MIDI-sized blot using a single antibody or condition. There's another that will allow you to process up to 2 mini blots at once using the same or different antibodies or conditions. Or you can process 6 vertically cut strips using up to 6 different antibodies or conditions. The Invitrogen Bandmate automated Western blot processor offers another option for automating the steps of the Western blotting immunoassay. The Bandmate Western blot system allows you to load the instrument with your reagents, select a program, and hit go. You can do other activities while the Bandmate automatically performs all the steps of the Western blot processing. The Bandmate can be a higher throughput option because it allows for processing of up to 4 mini blots or 2 mini blots at a time using different antibodies. It has flexible programming that will allow you to select a default standard Western blot protocol, or you can customize a program based on your preferences. The Bandmate works with your standard Western blotting protocol with no need to re-optimize conditions or use special reagents. The Bandmate allows you to process each blot with a different antibody, and collection tubes can be used to recover your antibodies for reuse in the future if desired. The automation of the Western blot processing improves consistency in your Western blotting results by precisely controlling the timing of all of the processing steps and providing consistent rocking throughout those steps. As mentioned in the previous slide, the Bandmate processor allows you to choose from pre-programmed protocols, or you can set up a program to perform your favorite protocol. The Bandmate can be programmed to dispense volumes of wash buffer up to 75 milliliters for mini blots and up to 150 milliliters for midi blots, and each step can be programmed with up to 10 repetitions per step. The Bandmate rocking speed can be customized between 5 and 40 cycles per minute, and incubation times from 1 minute to 96 hours can be programmed for blots that require incubation overnight or for longer periods. And the Bandmate can be used at room temperature, or the entire system can be placed in a cold room for long-term use if desired. The Bandmate system provides more consistent and often better results than manually processed blots. The images on the right show a comparison of a blot processed manually versus a blot processed using the same conditions and protocol on the Bandmate processor. In this example, 3 target proteins were fluorescently detected on an iBind imaging system. These results show equal to superior detection of all 3 target proteins on the Bandmate versus manual Western processing. The Bandmate system improves blot-to-blot consistency by precisely timing the block, wash, and incubation steps of the Western immunoassay. In these results, you can see the consistency of fluorescent detection of 3 different antibody targets in identical Western blots processed in 4 separate Bandmate incubation trays. In summary, there are a number of steps you can take to improve reproducibility and consistency in your Western blot results. Choosing precast gels and ready-made consumables with rapid transfer devices can reduce hands-on time, handling inconsistencies, and it can reduce your time to results. Using more automated tools for Western blot processing steps can also help reduce hands-on time and improve blot-to-blot consistency to help you achieve the best publication quality data possible. Now I will shift focus to special considerations for quantitative Western blots. As a result of the 2014 meeting held by the NIH and publishers, scientific publishers now have more stringent guidelines for what they wish to see from scientists who are publishing. Nature wants appropriate reagents, controls, and imaging methods with linear signal ranges to be used for quantitative comparison. The Journal of Biological Chemistry wants scientists to record how data were obtained, whether signal intensity was linear with antigen loading, and how protein loading was normalized. They also prefer signal intensity to be normalized to total protein loading by total protein staining rather than using housekeeping proteins. But if housekeeping proteins are used, they require evidence that manipulations do not affect expression. Manuscripts with quantitative Western results will likely come under greater review scrutiny. You can gain a better understanding of your data using our protein normalization workflows. In Western blotting, variability is usually due to unequal protein sample concentration, inconsistent sample loading, or variation in the transfer step. Sources of inconsistency can be monitored with visible or fluorescent gel and membrane-based labeling methods, followed by quantitation of total lane protein or with exogenous loading controls. Sample consistency and health can also be evaluated using internal housekeeping protein controls such as GAP-DH, beta-tubulin, beta-actin, or cyclophilin B. In the results on the right, you will see an example of a helolysate expressing RB1, which was serially diluted, separated, and transferred to a nitrocellulose membrane. The blot image on the left was stained with Invitrogen no-stain fluorescent total protein stain, and the protein of interest was probed with a primary antibody labeled with Alexafluor 650 dye. Normalization was performed for total protein and RB1, and the values were plotted against protein load. Housekeeping proteins are the most common normalization controls. The table on the right shows a list of standard housekeeping proteins and their corresponding molecular weights. It is assumed that housekeeping proteins are expressed at the same levels across experiments. However, recent studies have shown that expression of housekeeping proteins can vary across different cell types and biological conditions. Therefore, some scientific publications and funding agencies now require other forms of normalization or validation of normalization controls to ensure reproducibility and accuracy of quantitative Western blotting results. Total protein normalization is the preferred method because it's not dependent on a single loading control protein. The abundance of the target protein is normalized to the total protein amount in each lane. This improves the precision down to as low as 0.1 microgram of total protein per lane and improves data reliability and quantitative Western blot results. Invitrogen no-stain protein labeling reagent is an excellent choice for total protein normalization. No stain can be used for labeling proteins on your protein gel of choice, and it can be used to label proteins on nitrocellulose or PVDF membranes post-transfer. The no-stain reagent offers an easy-to-use protocol with mix and add reagents and can label proteins within 10 minutes. The resulting protein staining with no stain can be visualized with a range of imagers from UV, green or blue LED, or fluorescent light sources at a wavelength of 488 nanometers, such as the 488 nanometer excitation source on the iBrite FL1500 imaging system. No stain reagent provides accurate total protein normalization over a broad linear range for detection from 1 to 80 micrograms of total protein and has the sensitivity to detect down to 20 nanograms per band. No stain signal is stable and compatible with downstream chemiluminescent and fluorescent antibody detection. No stain reagent yields improved accuracy for normalization compared to housekeeping proteins. In this example, helolysate was stained with no stain reagent and compared with 3 different standard housekeeping proteins: beta-actin, GAP-DH, and alpha-tubulin. You can see that the signals from the housekeeping proteins get saturated with increasing protein loads of helolysate, while the signal from the lysate labeled with no stain reagent stays linear with increasing lysate loads, thus making the total protein normalization a more accurate way to normalize protein. Using no stain reagent for total protein normalization provides excellent concordance with protein load. Here you can see a comparison of different normalization methods. The percent error for no stain reagent versus the 3 standard housekeeping proteins from the predicted responses were computed and compared as shown in the table. The relative inconsistencies were normalized to the 10 microgram load. The signal measured would equate to the predicted signal, leading to the percent error for the 10 microgram loads being 0. The larger the percent error, the farther the signal is from the predicted response, resulting in reduced accuracy. The accuracy of the total protein normalization with no stain reagent was significantly greater and more reproducible than the protein normalization using all 3 housekeeping proteins. Compared to other in-gel total protein normalization methods, total protein normalization with no stain labeling reagent allows you to use your gel of choice without the need to change anything. And there's less risk of gel tearing with no stain reagent because there's no need to handle fragile gels after electrophoresis to expose them to UV light in the labeling reaction. No stain reagent is also versatile and can be used to stain proteins on membranes as well as gels. No stain provides a strong signal due to binding to plentiful lysine residues, and no stain does not require destaining like some other membrane-based total protein stains. No stain offers flexible detection and works with UV, green, or blue LED and 488 nanometer fluorescent channel imaging, so it doesn't require the ability to read an IR or near IR channels. You can streamline your total protein normalization by pairing no stain reagent with the Invitrogen iBrite imaging system. The universal mode of the iBrite imaging system makes it easy to capture images of gels and membranes labeled with no stain reagent, and the iBrite imaging system offers powerful integrated software to streamline the total protein analysis using no stain reagent labeled gels and membranes. No stain reagent and iBrite imaging system offer flexible, rapid, and accurate total protein normalization for your quantitative Western blot results. Choosing the right ECL substrate is also important for quantitative Western blot analysis. Chemiluminescent substrates are pushing the boundary of sensitivity, which can be very important for detecting low abundant proteins. However, for high or medium abundant targets, ultra-sensitive substrates may contribute to signal saturation, making quantitation virtually impossible. On the other end of the spectrum, standard ECL substrates may not be sensitive enough to detect mid to low abundant protein targets. Quantitative chemiluminescent Western blotting requires an optimal substrate that provides sensitive linear signals over a broad range of protein concentrations. Supersignal Westura is often the ideal choice for quantitative Western blot results because it has a long, stable signal that works across a range of target concentrations from low to high abundance. These blot results with Supersignal Westura show the importance of choosing the right chemiluminescent substrate to ensure you stay in the linear range of detection for your protein target of choice. To achieve the most quantitative Western blotting results possible, avoid overloading your sample and avoid overexposing your images to stay in the linear range of your protein target. Protein normalization is typically more quantitative than housekeeping proteins and is the preferred normalization method. Digital imagers are preferred for ease of use and quantitation because they have built-in tools like the Smart Exposure technology in our iBrite imagers that minimizes overexposure risks to help ensure you stay in the linear range of your protein. Imagers also provide software for normalization calculations, and for the best quantitation, select a chemiluminescent substrate with long signal duration that will help you stay in the linear range of detection for your protein of interest. In this slide, I'll briefly summarize a few key takeaways from all 3 webinar sessions that have been presented for achieving publication quality Western blot data. Because more stringent review guidelines have been developed in recent years, it is necessary to improve reproducibility and quantitation of Western blotting results and minimize digital processing of data. The steps we've reviewed throughout this webinar series include choosing a trusted antibody source and validate your target and verify with positive and negative controls. Choose from our many precast gels with optimized gel chemistries, buffers, and membranes to obtain the best separation and improve band resolution and background for your protein of interest. Improve detection of low abundance targets by choosing the right gel chemistry and using larger format wells. Choose a highly sensitive chemiluminescent substrate and consider using signal enhancers when studying very low abundant proteins. And use a digital imaging system such as the iBrite imaging system with smart exposure to obtain sensitive image detection. Increase reproducibility and minimize time to results through the use of instrumentation with pre-made consumables such as the iBlot-2 transfer system and transfer stacks, and improve consistency and hands-on steps by utilizing instrumentation like our iBind Flex system or Bandmate Western blot processing system to automate the Western blot processing steps. And last but not least, use total protein normalization instead of housekeeping proteins to obtain publication quality quantitative Western results. Invitrogen offers an online tool that can help you optimize your Western blotting experience. The Invitrogen Blot Builder Western blot product selection tool is designed to help you select the optimal products for your Western blotting needs by answering 5 questions about your protein of interest. When the 5 simple questions are completed, you'll receive a list of recommended products and a personalized Western blotting protocol. You can scan the QR code on the right or visit the URL thermofisher.com/blotbuilder to access the Blot Builder tool. Thank you so much for attending our webinar today. I believe we will be opening things up for questions now. Thank you very much.
And thank you, Kelli, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Joining Kelli will be Jordan Thompson, a product manager, electrophoresis from Thermo Fisher Scientific. He presented the part 1 from our webinar series. Welcome, Jordan. So Kelli and Jordan, let's take a look. We already have some great questions coming in from our audience. Our first question, what is the difference between dry and semi-dry transfer?
Hi, thank you. I can take that question. So the dry transfer is a unique transfer product that only is possible with Thermo Fisher Invitrogen select stacks for the power blotter or the iBlot-2 transfer stacks. The iBlot-2 and the select stacks actually utilize a unique gel matrix for the cathode and anode buffer. So there's no extra buffers that are required, and very little liquid is actually generated during the transfer step, as opposed to semi-dry transfers that, while they use less liquid than a tank transfer, do still require the use of a prepared transfer buffer to pre-wet all of the stack materials.
Thank you so much. And does the iBand Flex system require optimization, or can I use it with the same protocol I use for my traditional Western blots?
That's a great question. So there is some optimization of your primary and secondary antibody often required because the iBind system utilizes less liquid volume than traditional Western blotting protocols. You may recall from the presentation, the iBind Flex system uses sequential lateral flow to perform all the steps. So there's no timing or shaking or any of that. The blocking, the antibody binding, and all the washes occur automatically and sequentially. So in less than about 3 hours, you're ready to process your blot for final detection. And of course, you can still continue to use the same primary and secondary antibodies and, you know, your detection of choice.
Thank you so much, Kelli. And again, I want to thank our audience for these great questions coming in. Our next audience member says, can I use the Bandmate system for overnight incubations? Yes,
of course. The overnight incubation is entirely possible. The Bandmate can be set up in a cold room for long-term use, or overnight incubations can actually be performed at room temperature. Our R&D scientists have shown equivalent or often better results in Westerns performed at room temperature overnight versus overnight in a cold room or at 4C. I do know that often people that are collecting their primary antibodies for reuse prefer to have the instrument set up in their cold room, but it is entirely possible to do those incubations overnight at room temperature as well.
Thank you very much. Our next question, can dry and semi-dry transfer be used for transferring high molecular weight proteins like BRCA 390 kDa?
So yes, the system, both the the iBlot and the Power Blotter system can transfer high molecular weight proteins. It's often better to use something like a tris acetate gel that is specifically designed for high molecular weight proteins, and you may need to transfer just slightly longer, so maybe up to 10 minutes for an iBlot, for example, will help you get more of that high molecular weight protein out of the gel and onto the membrane. But it is, yes, entirely possible to use for high molecular weight proteins.
Thank you, Kelly. And we have time for a few more questions. Can no stain be prepared as a master mix, or does it need to be made fresh right before using?
I can take that one. No stain can actually be prepared while you're doing your separation or your transfer, so it's actually really easy. You can make a master mix. It's stable for at least an hour, and you can scale up the amount or the volume of it so that you can prepare what you need for all the gels or all the membranes that you're labeling at a single time.
Thank you, Jordan. And how does no stain label proteins in a sample?
So no stain actually uses, sorry, Kelly, I'll take this one again. No stain actually uses a fluorogenic molecule that gets activated. So there's three components to the no stain kit. It's a labeling buffer, an activator, and the fluorogenic derivatizer. And so in the presence of the activator, the derivatizer actually reacts with primary amines and creates which is traditionally the lysine side chains, and it creates a covalently and highly fluorescent product that can be visualized on imagers, such as the iBind.
Thank you, Jordan. And did you want to add anything to that, Kelly?
No, I think Jordan covered it well. Thank you.
Perfect. Our next question, how to successfully transfer high molecular weight proteins, such as 150 kDa?
Yeah, so again, it's highly, possible to transfer high molecular weight proteins using semi-dry or dry rapid transfer. Often you'll get better results if you use gel like a tris acetate gel, but even a regular gradient gel can transfer a high molecular weight protein quite efficiently using any of our systems. Often you need to transfer just slightly longer, but you can get very good results.
Thank you so much, Kelly. And I want to thank you for your responses and your presentation today. Did you want to provide any final comments to our audience before we go?
Yes, so I would like to thank everybody again for attending the webinar series and for attending today's session. And I hope that you found the information helpful and will be able to get better quality Western blotting results as a result of listening to the 3 webinar series. Thank you so much for your time, and I think that's it.
And we want to thank you again, Kelly, for your time today. And I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, I'd like to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and stay safe. Bye-bye.